Today's topic, SC Framework, the Quality Control Notebook. If you haven't watched the video about the first notebook, we recommend you to go back and watch this video first. In this notebook, we focus on the quality control of the data. The aim is to reduce the dataset to meaningful cells and genes. In this example, we focus on droplet-based sequencing methods, but the same parameters are also applicable to other methods such as plate-based. This notebook can filter low-quality droplets or wells, low-quality cells, and filter ambient RNA to only work on high-quality data. To ensure those quality standards, we focus on three key aspects. The number of counts per barcode, also called count depth, the number of genes per barcode, and the fraction of reads derived from mitochondrial versus nuclear origin. Outliers based on these covariates are filtered out. Such outliers may occur if a cell is dying, leading to broken membranes that cause mRNA leakage, resulting in low count depth and detected genes, along with elevated mitochondrial counts. It is important to note that these covariates can vary between cell types. For example, cells involved in respiratory processes may have a higher mitochondrial content compared to other cells. Coming now to the notebook. As we did in the previous, we start by loading all the required packages for this notebook, as well as the configuration file containing all in and output path for the analysis, and show the version of the main packages. Then we load the anndata H5AD file generated in their respective predecessor. Here it is, the output of the assembly notebook. This and all following notebooks will display the anndata object with the OBS and the var table to get an overview of the data. Next, we start section 4 by filling out some general metadata and filtering criteria for the dataset. This starts by setting the species of the organism from which the samples were taken. In our case, this must be set to zebrafish. Then we set the condition column. This refers to a column name in the OBS table that contains the condition value of the cells. In this case, we aim to determine if gene expression changes during post-fertilization development, so our condition is time point. The SC toolbox expects gene names to be in the index of the var table. If gene names are in a different column, set the variable gene underscore column to the respective column name. The min underscore genes parameter is typically set to 1. This sets the minimum number of genes that a cell needs to express to be kept for the QC analysis. This filtering step is one of the first steps done. Our more strict filtering is done later in the notebook. Next, we set the options for the doublet prediction. You can turn off the doublet filtering by setting filter underscore doublets to false. The threads parameter sets the number of CPU threads available for multithreading. The rule of thumb here is more threads equals faster runtime. With the doublet underscore threshold option, you can set a specific threshold for the doublet prediction. If it is kept none, an automated threshold will be set. The use underscore native parameter lets you enable to run the prediction via the native scrublet package. By default, the scampy method is used. With the following two parameters, options for the sex prediction can be set. You can turn off the sex prediction by setting predict underscore sex to false. The sex prediction estimates the sex of each sample using female-specific marker genes. These genes vary between organisms, thus the gene can be set with the female underscore gene parameter. For zebrafish, we set it to the RBPMS2A gene. The notebook will estimate upper and lower thresholds for QC metrics. You can later on accept the defaults or adjust them manually. With the next parameters, you can specify whether thresholds are estimated per condition or globally across all data, and which function should be used for the automatic threshold selection. Options include Gaussian mixture model, median absolute deviation, or a custom function. With the fun underscore KWERGS parameter, you can set additional parameters of the filtering functions. We recommend new users to stick to the default parameters. Next, we set the parameters for gene labeling and gene group filtering. The SC toolbox uses internal gene lists or identifies genes by name, for example MT, for mitochondrial, RPS, for ribosomal genes. With the highlighted parameters, you have the option to choose from the SC toolbox provided internal lists or give a path to a text file with a custom gene list. If this option fails, the SC toolbox tries to label genes based on a given regex. This regex can also be set here. The filter underscore mito, filter underscore ribo, and filter underscore gender parameters lets you turn the respective filters on or off. In this case, we want to filter for mitochondrial and ribosomal genes. The dataset contains only female samples, thus we do not care about removing those genes. If the pre-processing was done via Starsolo, you can add the path to the quant folder. With this set, additional QC plots from the Starsolo data will be plotted. The next two parameters can be set if ambient RNA should be removed. This requires a raw, unfiltered anndata object containing all droplets. To enable this filtering step, insert the path to the raw H5AD into path_raw_anndata. The epochs parameter sets the number of epochs. This sets the number of iterations for the denoising algorithm. 150 epochs is recommended, and we advise not setting it below 100. Lastly, the overwrite parameter lets you overwrite previously applied SC toolbox filtering steps. Set this to true if you want to apply new filtering on top of previously applied filters. After selecting all options, we can start running the actual filtering. This starts with a cell that ensures that the condition column, in our case, time point, is of type category. This is required by some of the functions used in this notebook. Section 4.1 creates the Starsolo quality plots if the quant folder was given earlier. In Section 4.2, we label genes based on their general associations using the parameters configured earlier. Since the SC framework does not provide internal gene lists for zebrafish, we use the fallback option to identify genes via the specified regex patterns. For this dataset, we successfully identified 37 mitochondrial and 97 ribosomal genes. As no list of gender-specific genes was provided, no gender-specific genes are labeled in this analysis. The resulting labels are stored as columns in the anndata.var table. Next, we calculate a variety of QC metrics for cells and genes. Those are the metrics we mainly filter on. We will discuss each metric later on in the video. Then, we predict and remove doublets. First, we remove cells with a gene count lower than what we set with min_genes earlier. With min_genes set to 1, we filter out empty cells, which means cells with no gene expression. Doublets, also called multiplets, are cell barcodes that are assigned to 2 or more cells. This happens when multiple cells are encapsulated in the same space, droplets or wells, during the separation stage. Therefore, a doublet is an artificial cell. It is an accumulation of the expression profiles of multiple biological cells, potentially skewing results due to high read counts. Therefore, it is recommended to remove doublets. We use the Scrublet tool to predict and remove doublet events from the dataset. Scrublet predicts doublets by scoring each cell based on the similarity to simulated doublet cells. A threshold applied to the distribution of doublet scores predicts the cells to be singlets or doublets. Scrublet can either infer a threshold based on the doublet score distribution when the parameter is set to none, or the user can set a manual threshold. A manual threshold should aim to be in the valley between the first and second peak. In our case, we set the threshold to 0.2. In the following input cell, you can again decide if you want to remove the predicted doublets or if you want to keep them, followed by the cell that then removes the doublets if enabled. With the threshold set to 0.2, we filtered out 1,278 potential doublet cells, resulting in 44,716 cells retained from the original 45,994 cells. If the sex of the samples is uncertain, the notebook provides an option to predict the sex using the previously specified female marker gene. In our case, the set female marker gene is expressed in all samples, as indicated by the red background in the violin plots. A red background signifies that the function predicted the sample as female, while a white background would indicate a male prediction. Since all samples in our dataset are predicted as female, this eliminates the possibility of a sex bias in the data. Consequently, further sex-related corrections or gene removals are unnecessary for downstream analysis. Next, we continue with the cell filtering. Cell filtering is a critical step in this notebook. Typically, some cells are of low quality, which can introduce noise and compromise the accuracy of downstream analysis. The goal here is to reduce this noise by filtering low-quality outliers based on the quality metrics calculated earlier in this notebook. The section starts by giving you an overview of the cell metadata, which now includes the quality metrics. Here you can see an overview of the commonly available metrics, which includes the number of genes associated with a barcode, the total amount of reads detected for each barcode, the percentage of mitochondrial reads per barcode, and many more. In the following input field, we can choose on which metrics we will filter the cells and if the initial threshold should be automated or not. You can also choose if the threshold estimation should be done globally or individual per condition. To choose which metrics to use, add them to the default_OBS_thresholds dictionary. For an automated estimation, set min and max to none, but you can set the values to your liking. We recommend to use the automated version, as we do here, since you can adjust the threshold manually later on. In this analysis, we use the number of genes, the total amount of reads detected, and the percentage of mitochondrial and ribosomal reads per barcode as metrics for filtering. With the initial threshold set to none, the next cell automatically estimates initial thresholds using the algorithm chosen earlier. Here we use the Gaussian mixture model estimator, which identifies the main component of the distribution and defines the threshold as 3 times the standard deviation from the main component's median. If thresholds are set manually, the table will show the user-defined values instead. The next plot visualizes the number of cells filtered out by individual quality metrics, based on the thresholds in the table above and the overlap between pairs of metrics. This visualization helps quantify the contribution of each metric to the filtering process. For example, if n_genes filters 5,000 cells and PCT_count_ is _mito also filters 5,000 cells, but their intersection, so cells filtered by both, is also 5,000. This would indicate that the two metrics are targeting the same low-quality cells and thus have identical filtering impact. Conversely, if the intersection is smaller, more cells are filtered. This means that the metrics are removing distinct subsets of cells, highlighting their complementary roles in quality control. Our next step is to adjust the thresholds for cell filtering. The next cell displays an interactive plot featuring violin plots grouped by the condition_column parameter we defined earlier. Red dotted lines indicate the thresholds. On the left side of the interface, you'll see the quality metrics specified in the input cell. By selecting a metric, you can adjust its minimum and maximum thresholds using a slider. The goal of this step is to remove low-quality cells while preserving biologically relevant populations. When interpreting the violin plots, focus on excluding outliers, as most high-quality cells cluster in the main body of the violin while balancing the technical and biological context. For example, heart samples may intentionally retain cells with higher mitochondrial content. For rare cell types, for example, small clusters use less strict thresholds to avoid overfiltering. For this dataset, we will set the thresholds to the shown values. The next 3 cells will display a table of final thresholds for cell filtering, a plot showing the number of cells filtered using these thresholds, and a pairwise comparison of the thresholds applied to each quality metric. These visualizations provide insights into the impact of each threshold and help assess their biological relevance. For example, the pairwise comparison highlights whether thresholds for different metrics overlap significantly or target distinct subsets of low-quality cells. Since filtering has not yet been applied, you can return to the interactive plot to refine thresholds if needed. For this analysis, we will proceed with the final thresholds as defined. With the next cell, we apply the set thresholds to the dataset. In this case, we filtered out 10,462 low-quality cells, retaining 34,254 high-quality cells in the dataset. Running the next cell generates violin plots for each quality measurement, showing the cell distribution after filtering. With all low-quality cells removed, we proceed to remove low-quality genes from the dataset. Before doing so, we must first recalculate the QC metrics, as the previous step removed cells that could influence gene-level quality assessments. Next, all genes with no expression across any cell are again removed from the dataset. This step is necessary to ensure compatibility with downstream analyses, such as differential expression or clustering, which require at least some expression in the dataset. Then, similar to the cell filtering, The notebook gives you an overview over the gene metadata, which now includes the quality metrics, before you can select on which metrics the genes should be filtered. In this analysis, we use the default metrics, the number of cells that contain reads associated with the gene, and the mean amount of reads over all cells. By default, all genes are included by setting min and max to -infinite and infinite. Here you can see a list of commonly available metrics, but any numeric column in the var table can be used. With the initial thresholds set by the next cell, we can continue with the manual adjustments, which works just like we did it with the cell filtering. For our dataset, we set a minimum threshold of 100 cells for the number of cells in which a gene is expressed and apply no upper threshold. This means all genes expressed in fewer than 100 cells will be excluded from the dataset. While the goal is typically to retain all genes for comprehensive analysis, filtering out low-expressed genes can be beneficial in specific cases. On one hand, to reduce noise, low-expressed genes often represent technical artifacts or insufficient read coverage, and on the other hand, improve the downstream analysis. Removing these genes can enhance the quality of clustering and dimensionality reduction by focusing on biologically relevant signals. After setting the threshold, you will get a short overview from which you can go back and adjust the thresholds again before applying the filtering. For this dataset, we filtered out 10,444 genes, retaining 17,959 genes in the dataset. The final distribution violin plots of the quality metrics are then plotted by the next cell. The final gene filtering step involves removing mitochondrial, ribosomal, and gender-specific genes based on the parameters defined earlier. Since our dataset contains only female samples, we exclude gender-specific genes from filtering and focus on removing ribosomal and mitochondrial genes. This results in the removal of 25 mitochondrial and 95 ribosomal genes from the dataset. The last and optional step of this notebook is the removal of ambient RNA. Ambient RNA can introduce unwanted technical artifacts in downstream analysis, particularly in single-nucleus datasets where ambient RNA contamination is more prevalent. To address this, the next cell employs SCAR, a tool that estimates the ambient RNA profile by averaging cell-free droplets and uses an autoencoder neural network to correct the count matrix. However, since we are working with single-cell data, where ambient RNA contamination is typically negligible, this step is not required and will be omitted in our analysis. As for all individual notebooks, the last step is to give a short overview over the final anndata object and to save the anndata object into an H5AD file. With this finished, we have completed the second notebook of the RNA analysis. If you plan to use this or one of our other notebooks, please check out our Docker image, which comes as a full functional environment for the SC framework to be used within a web browser. The next video will focus on the normalization and batch correction of the data. If you need further assistance or want more information, be sure to check out our documentation or visit our GitHub repository. Thanks for watching.